Welcome to the recording for the Unit 2 procedure, or the lab, instructions. And we'll also discuss this idea of collision theory, which will be important for your kind of understanding of the lab. But in the Unit 2 lab, we investigate different ways of getting salt to dissolve faster. So we're going to learn about collision theory. We're also going to discuss how this will relate to your Unit 2 lab. Oops, that's in the place. So, Unit 2 lab, there are 3 parts. In your kits, you should have some bottles that look like these that have 3 different types of salt in them. One is going to be pellets, so like big chunks of salt. One is coarse, which kind of looks like kosher salt. It's smaller pieces, but bigger pieces than the fine salt, which is like your typical baking salt or table salt. So one thing is, if you happen to run out, you'll probably be able to replace potentially these with stuff you have at home. So part 1 is the boring part. You're going to take a pellet, put it into one vial. So these are some vials that you likely have in your kits. All right. Oops. And then fill them up to the 20 milliliter mark. So there are markings on the side, and actually it looks like I filled up mine even further. But whatever you do, fill all 3 of them up to the same amount. Then one vial you're going to put the pellet, and the next vial you're going to put 4 grams, and you have an electronic balance in your kit. 4 grams of the coarse salt, and then the third vial, 4 grams of the fine salt. All right, and then here you're just going to let them sit. Don't do anything. Don't shake it. Don't mess with it. And let it dissolve for up to 45 minutes. For this one, you'll probably have to go the full 45 minutes because particularly that pellet probably won't dissolve, if any of it. And then record some observations. You know, how much of the salt is left after that 45 minutes. All of it, some of it, something like that. Part 2 is going to be set up the exact same way as part 1, except this time, instead of just letting it sit there, all right, every minute that passes on the timer, I want you to take your vials and shake them for 5 seconds. And then put them down, let them sit for a minute, and then shake them for 5 seconds, let them sit for a minute, shake them for 5 seconds. And keep doing that until either everything dissolves, record what time it is when it dissolves, or after 45 minutes, if something still isn't dissolved, then just record how much is left. All right, and actually for this one, you're fine just going 30 minutes. All right. Or if everything dissolves in less than 30 minutes, you can stop recording at that point too. And part 3 is set up a little bit differently. You're going to have 3 vials, or even maybe 3 glasses from around the house. But in all 3 of them, you're going to put the same amount and same type of salt in. All right, so this is going to be a little dependent on what you have left in your kit. So like if you have more fine salt left, use fine salt. If you have more coarse salt, use coarse salt. Or if you run out of everything, then use whatever type of salt you have lying around the house. But make sure that in all 3 vials, you're putting in the same size salt, the same amount. And the difference is going to be whether or not your water temperature is hot, room temperature, or cold. All right. Now, one thing that I recommend, because this is going to help you get really easy to interpret data, is when it goes to the hot and cold water, go with extremes. All right. So like for your cold water, before you get going on the lab, take a cup, throw some ice in the water so that the ice can melt, and then you're going to have super cold water. There's also, when you're getting your hot water, let your faucet run, get as hot as it can get, use the hottest water you can get out of your faucet. All right, and then room temperature water, obviously, is just, before you get, even at the beginning of the lab, just take a glass of water out and get that at room temperature. But make sure your temperature differences between your vials are as extreme as you can make them, because that'll make it really easy for you to kind of tell the different times at which dissolving will occur. Now, once you get this set up and you have your hot room temperature and cold water, then you're going to do this very similarly to part 2 of the lab. So shake everything for 5 seconds, let it sit for a minute. Shake everything for 5 seconds, let it sit for a minute. All right? So if any of this isn't making sense, let me know. I can re-explain. But these instructions that I'm describing here are also in your lab manual. All right, so you can follow along with the setup in the lab manual. That way you have nice written instructions. This is just meant to be an overview of what that lab manual is saying, and hopefully clarifying things if the written words don't quite make sense. I don't know why this says assessment, but here are some example tables that you might want to collect. I do highly recommend recording times, all right, at which things dissolve. If after, the 45 minutes or the 30 minutes, things are still left, then just record a general observation, like half the pellet is still there, or most of the pellet is still there, half the amount of fine salt is still there, something like that. All right, so you should either, if it completely dissolves, have a time at which that occurs, or a description of how much salt is left compared to what you started with. All right, you need these for your lab report. All right, many of the points on the lab report are going to be tied to sharing your data, so make sure you record these somewhere. And then, of course, pictures, so turn in your results. All right, you can take pictures like this, or if you create those lab tables, turn in pictures of your lab tables. All right. Either way, it'll give me an idea of what's going on, if the lab was successful for you, that sort of thing. But at this point, I want you to make some predictions. What do you think is going to happen? All right, so based on our 3 parts of our lab, we've got a few things that we are kind of comparing. We've got the 3 different sizes of salt. So we can use a couple of either part 1 or part 2, or both of them potentially, to kind of see what happens based on size of salt. All right, so what do you think is going to dissolve the fastest? The big chunk pellet, the coarse kind of medium-sized salt, or the fine super tiny salt? All right. The other thing that we can observe or compare in this lab, if we directly compare part 1 to part 2, is agitation or motion. All right, if we shake the vials, what does that do? Is shaking the vials going to cause dissolving to happen faster, or is leaving them still? So that's another prediction that you can make. And then the last thing in part 3, what we're really testing with that one is temperature. So which temperature should get the salt to dissolve fastest? Hot water, room temperature water, or cold water? All right, so go ahead, think about this for a second, make some predictions. All right, some of these probably you might even already know, but predict what you think is going to happen. All right, so the concept behind this, and we've started talking about this in a sense, because we talked about some trends that we already know lead to faster dissolving, so like having more surface area, combining polar solvents with polar solutes. For solids, we know temperature makes things dissolve faster, right? But really what's going on with all of this is the idea of collision theory. And I pulled this right out of the lab lesson, all right? So the knowledge that dissolution can be explained in terms of particle collisions is called collision theory. This theory states that the particles that make up any solute or solvent are in constant motion. When solvent particles collide with solute particles, dissolution is the result. All right? So ultimately, to get dissolving to happen, in our case, our solute is the salt, our solvent is the water. The salt and the water molecules need to collide. All right? When that happens, all right, we get this pattern of group of water molecules surround a salt molecule. A group of water molecules surround another salt molecule. And eventually, that happens so evenly that you don't see the salt anymore. All right? But the only way this happens is if they actually come into contact with each other. They have to collide. So, I want you guys to just think about when you've seen collisions take place. All right, maybe you can think of, what happens when a car accident happens, or say if you've ever been bowling and you see the bowling ball hit all the pins, that sort of thing. All right, so if you just kind of visualize this whole collision taking place, if you think about it, when things bump into each other, it usually causes stuff to break up. And that's going to be the exact same thing that happens inside our salt water. All right, you can think of it as even the fine salt is going to be multiple. If you remember Avogadro's number, all right, we're talking if you have a mole of salt, which is about 60 grams, no, more than that, 75-ish grams. And even if we're only dealing with 5 grams, we're still dealing with, 6 times 10 to the 22nd salt molecules, all right? So even a teeny tiny little fine pelt salt is still going to be a whole bunch of salt molecules associated with each other. So when that water molecule collides with it, it's going to break it apart, okay? So you made some predictions. I've done the lab before. I also know these would have been my predictions as well. All right, so if everything goes according to plan, those 3 questions that we had, size, fine salt should dissolve the fastest. When it comes to motion, agitation or shaking should make things dissolve faster. So part 2 should dissolve faster than part 1. All right, and in part 3, your hot water should dissolve things pretty quickly. So now the trick is, and we will continue to discuss this further, but the trick is to kind of think about, well, how does this, how is fine salt able to provide more collisions than, say, pellet salt? All right, and this is going to combine what we've already discussed with collision theory. All right? So what I want you to do is consider the pellet salt to be our cube, and the fine salt to be all the little pieces separated from the cube. All right, water has an easier time coming in contact with all of the fine salt particles, much more so than the pellet salt. All right, so you've got more surface area means more collisions. All right, so that's kind of the idea here. So for each of these, you want to be able to, describe how does the faster dissolving condition cause more collisions. All right, we will talk about this in more detail when we get to your lab report. But, think about these other 2, because we will talk about them, and I want to get you guys explaining these ones.